lecture, we'll talk about the last topic of the chapter molecular basis of inheritance, that is 6.10 DNA fingerprinting. So, we reach the last topic of the chapter, that is 6.10, and the name of the topic is DNA fingerprinting. It is DNA fingerprinting. Don't think that so the fingers DNA will be taken out and then they will decode it. It's not like that. So we'll try to understand why did they name the technique as DNA fingerprinting. Okay, so before this technique there was another technique which was developed in West Bengal for identification of criminals in the year 1897, right? So that technique is called as dermatoglyphics. What is the name of the previous technique? So it is called dermatoglyphics. Right. Now, what is dermatoglyphics? Dermatoglyphics is a science of studying the fingerprints, literally the fingerprints. Dermatoglyphics is a science of studying fingerprints. That science of studying the fingerprints is called as dermatoglyphics, and this technique, dermatoglyphics, was developed in 1897 in West Bengal. This technique of dermatoglyphics was developed in the year 1897 in West Bengal. It was developed in West Bengal. So, this technique has been developed for criminal investigation. If you want to identify the criminals, then this technique will help us because they leave their fingerprints here and there, they leave their fingerprints. With that, we can trace out the person. That technique where we are finding out, where we are studying the patterns of ridges, right? So, what is dermatoglyphic? It is a technique. It is a technique where we study the patterns of fingerprints. We study the patterns of fingerprints. We study the ridges. We study the cones and lines. Where are they present? Present on the skin of, where are these lines present? They are present on the skin of fingers, palms and soles, right? So, if you check your fingers, on your fingers, you can see some lines, you can see some cone shaped appearance, some circles we can see, some ridge type of structures we can see. So, when we study those fingerprint patterns, what is that science called? That science is called dermatoglyphics. Derma means skin, study is called, study of patterns is called glyphics, right? So, it was developed in 1897 in West Bengal for criminal investigation, right? So, each person has unique fingerprints. That's how you can identify, right? So, each person, they have unique fingerprints. So, which will help us to identify that particular person. But then, so we can misguide also, right? So, we can wear gloves, so where the fingerprints are not, will not appear. That is one way. Or we can surgically change the fingerprints also. So, this method is not proven effective very much. Why? Because fingerprints can be covered. That's one way. And the second way is surgically, so we can change the fingerprints also, which will misguide. That is the second thing. Then scientists thought of developing a better technique which cannot be changed either by surgery or you cannot hide it. You cannot hide it, right? So, what is that technique? So, the next technique which came, which is better than this DNA fingerprinting. So, that technique is called DNA fingerprinting. So, the better technique which cannot be changed, which cannot be altered, Why it cannot be altered means DNA fingerprinting is altered. How can we alter it? By surgically we can alter it, but the DNA cannot be altered, right? So the better technique which cannot be altered was developed. You know who developed DNA fingerprinting technology? It was developed by Alec Jeffrey. Developed by Alec Jeffries. In which year? In 1984. So, DNA fingerprinting technology was developed by Alec Jeffries in the year 1984. And what are the other names we can tell? So, DNA fingerprinting is also called, what are the synonyms? DNA fingerprinting technology. It is also called as DNA profiling. So, what is the other name for this one? They can ask you like DNA, what is DNA profiling? It is the same thing. DNA profile or DNA profiling is also the same thing. Or it is also called genetic fingerprinting. What is DNA? DNA is a genetic material. It has a genetic information. So, we can also call this technique as genetic fingerprinting. Genetic fingerprinting. Who developed this technique, children? It was developed by Alec Jeffrey in the year 1984. Alec Jeffrey in the year 1984, and we told it's not like the traditional fingerprinting technique which can be changed or which can be altered or which can misguide it. Whereas these DNA fingerprints, these DNA fingerprints are perfect way which help in forensic science, which help to trace, find out the criminals. And since these fingerprints are heritable also, so we can use it to find out the paternity. Mother is a fact, right? So mother gives birth to the child, so mother is fact. But the father, if there are any disputes in the family, if you want to find out whether the father is a biological father of that child or not, so we can see DNA fingerprints. A child, 50% of the child's DNA matches with the mother, 50% of the child's DNA matches with the father. So it is used in paternity testing, father testing. It is used in criminal investigation. It is used in evolutionary studies, right? So there are so many applications. We'll see it at the end of the session, right? So we understood what is DNA fingerprinting. DNA fingerprinting means identifying the DNA. Identifying the DNA, and you are using this identifying the DNA to compare with others. You are using for identification technique. And these are the other names of DNA fingerprinting. And it's developed by Alec Jeffrey in the year 1984. In the previous lecture, we discussed about human genome sequencing, right? In the previous lecture, we discussed about human genome sequencing, and there we told 99.9% of the base sequences among the humans are the same. Means my DNA and all of your DNA, how much we are similar with each other? 99.9% base sequences of all the humans are same. How do you came to know that? After doing the genomic sequencing, you came to know that, right? So 99.9% of human base sequences are same, or they are common for all of us. They are common for all of us. That means how much do we vary from each other then? Only 1%, only 1%, because 99, not 1%, 0.1%. We are telling 99.9% we are same, means that means 0.1% a person is having different DNA sequence from others. Do you agree? So, Human Genome Project, after completing Human Genome Project, the scientists have proposed the salient features. From there, we came to know one point, that 99.9% of the DNA sequence among all of us is same. Means in other way, you can tell that 0.1% we are different from each other. That means that 0.1% makes me unique from rest of you, right? So, I am very unique. My height, my weight, my behavior, so my pronunciation, everything is different. It's unique for me. So, what is that DNA sequence which is making me unique, which is making my phenotypic appearance unique, is 0.1%. So, 0.1% difference, 0.1% difference in the DNA sequence makes a person unique. It makes a person unique where? In phenotypic appearance, right? in phenotypic, if they tell, they give the same statement and here they'll tell genotypic appearance, so then it's wrong statement. NCERT says that this 0.1% difference makes every person, makes every individual unique. How? In the phenotypic, in the morphological appearance, in the phenotypic appearance. Understood? So let's see what we have discussed. Take a screenshot afterwards, then we can continue, right? So we came to the last topic of the chapter, 6.10, DNA fingerprinting technology. So before talking about DNA fingerprinting technology, let us talk about another science. What is it called? Dermatoglyphics. What is this science? It is the science of studying the fingerprints. When this technology was developed in 1897. Where? In West Bengal, which helped in criminal investigation. So what is this technique? In this technique, we will study the patterns of the fingerprints, patterns of the ridges, patterns of the circles, cones, lines present on the skin of the fingers, palms and the soles, right? And it also helps us in criminal investigation. It also helps us in identifying certain genetic disorders. In chapter number 5, principles of inheritance and variations, when we talk about Down syndrome, in Down syndrome, we'll tell characteristic palm crease is a symptom of Down syndrome. What do you mean by characteristic palm crease? If you see my palm, so here I have one prominent line, here I have another prominent line, and this is the third prominent line. All of us will be having these three lines, right? Little different from each other, but three lines we can easily see. One is this line, other one is this line, and the other one is this line. Now I am telling, in Down syndrome, there is a characteristic pattern. What is that called? Characteristic palm crease. Now in characteristic palm crease, what happens? You know, this line, the line on the top and this line will merge. Both of them will merge and like this. The second line and third line will merge and it will make a common line. It will make a common line. This symbol is called as characteristic palm crease. Means these fingerprints are also helping us to identify genetic disorders. From Down syndrome, we came to know that it helps us to identify the genetic disorders, right? But then the problem with this fingerprinting is they can be altered. Surgically, they can be altered, they can be covered, right? So then we want to develop a better technique. Then Eric Jaffrey came. He developed DNA fingerprinting technology. In which year? In 1984. What is that technique called? DNA fingerprinting technology. What is the other synonyms for this technique? DNA profiling. It can also be called as genetic fingerprinting. Now, in the previous session, we studied about the human genome sequencing. Through that, we came to know that 99% base sequence of all the humans is the same. Means to tell that 0.1% we are different from each other. And this 0.1% which makes a person unique in phenotypic appearance from the other person. Quickly take a screenshot. Right. Now, if one aims to find out the differences between two individuals, right? So a crime has happened, a murder has happened. So there are, so we recovered the DNA sample from that crime spot. Now there are two suspects. There are two suspects, suspect number A, suspect number B. So we have to prove it, whether this person has did it or the second person has did it, right? So then for that, what we need to do? Now we have to find out the genetic differences between these two individuals, right? We have to find out the genetic differences between these two individuals with the crime spot DNA. We have to compare it. Now, for that sake, if you try to do the sequencing of this person DNA, suspect A and suspect B, if you have to do the sequencing, so we have seen that how many years it took for the scientists to do the sequencing of one person, 13 years, how many countries, so many countries. What's the cost of that project? $9 billion US, isn't it? So now again 9 billion for one person, 9 billion for one person and 9 billion for the DNA which got collected and from the suspect, no one can bear it, right? So that is why it's a daunting task. It's a very difficult task. So if one aims to find out the genetic differences between two individuals, let us write on. If one aims to find out the genetic differences between two individuals. So this is the sentence of NCERT. genetic differences between two individuals. If you want to find out the genetic differences between two individuals, means you have to do the DNA sequencing of those two persons. Means we have to do the DNA sequencing. So, are you thinking DNA sequencing is an easy task? It's a very expensive task and it's a daunting task, right? So, which is very expensive and difficult task, right? So, then we can't afford it. We can't afford that amount of time, that amount of money. We can't afford it for doing the sequencing every time, right? So, then what is the other way to find out? But if we try to compare the two sets of genes instead of the DNA sequencing, instead of completely doing A, B, C, D, E, F, G, H, I, J, K, L till the Z of this person's DNA and this person's DNA, randomly cut it at some places and then just compare them, okay? Just compare them to at certain levels. So you can find out whether they're similar or dissimilar. So that's what we want to find out. We don't want to do the complete sequencing, right? We don't want to publish a paper on that. We just want to see, are they similar or are they dissimilar? For that, you need not study the whole thing. So that's why they have developed a new technique where we'll compare the sets of the genes, right? But if we try to compare the sets of the genes, if we try to compare the sets of the genes of two different individuals, right? Instead of completely doing the sequencing, if we just try to compare the sets of the genes of two individuals rather than DNA sequencing, it is an easy task. Instead of sequencing, if you just cut it and if you compare, it is an easy task. It becomes an easy task. It is an easy task and it is fast also. It is quick and fast, isn't it? Now, that technique of comparison of two DNAs is called DNA fingerprinting technology. Understood? So, DNA fingerprinting technology involves complete sequencing or it involves identifying the differences. Come on, tell me. DNA fingerprinting technology involves identifying the differences, right? In some places, right? Let us write down. So, we can tell that DNA fingerprinting technology. DNA fingerprinting technology, what does it involve? It involves identifying differences only, right? It will just identify differences, will identify differences in some special regions, right? In some special regions. Instead of doing the complete sequencing, we will try to identify the differences between the people at some special regions. Those special sequences are called repeated DNA sequences. What are the sequences now we are looking at the time of DNA fingerprinting? Repetitive DNA sequences, special regions in DNA sequence. We are looking at the special regions in the DNA sequences. Now, what are those special DNA sequences called? They are called repetitive DNA sequences. What are they called? Repetitive DNAs. Now, the name itself is telling. What do you mean by repetitive DNA? Repetitive DNA means it is repeating many times. That is why it's called repetitive DNA. Repetitive DNA means many times the DNA sequence gets repeated. Am I correct? If the DNA sequence gets repeated many times, then yeah, you can call it as a repetitive DNA, isn't it? Now, if you want to, you understood now what is repetitive DNA. Where is the repetitive DNA? In our DNA itself, in our genomic DNA itself. Now I want to separate it out also, right? So, a repetitive DNA, if you want to separate it from our genomic DNA is bulk DNA. Repetitive DNA is small. It is very small. If you want to separate the repetitive DNA from the bulk of the genomic DNA, what is that technique we need to use? It is called as density gradient centrifugation. Go with density gradient centrifugation technique. Then children, you can separate this repetitive DNA from the bulk of the genomic DNA. Then the bulk of the genomic DNA will give a bigger peak, and this repetitive DNA will make a small peak like a satellite. So it is called as satellite DNA. So now I used another word for repetitive DNA. What is that word I used? Satellite DNA. Why did I call the repetitive DNA satellite DNA? When I tried to separate the genomic DNA from the repetitive DNA by using density gradient centrifugation, the bulk of the genomic DNA has given a bigger peak, whereas the repetitive DNA has given a smaller peak like a satellite. So that is why it is called satellite DNA. If you want, we can write down like if we separate genomic DNA, if we separate genomic DNA from the repetitive DNA, if we separate genomic DNA from the repetitive DNA, by which technique? By, I told it is density gradient centrifugation technique. So we will prepare a density gradient column and we'll apply centrifugal force. Now the particles, depending on their densities, they nicely go and sediment there. So when we separate the genomic DNA from the repetitive DNA by density gradient centrifugation technique, when we do this technique, now the genomic DNA forms a big peak. So then the genomic DNA forms a big peak, right? And satellite DNA, how it will form? Not satellite DNA, repetitive DNA. Repetitive DNA, I told it forms a small peak. It will form a small peak. Since it is forming a small peak, it appears like a satellite. So that's why we are calling the repetitive DNA as satellite DNA also. Because it is forming a small peak, scientists, they told we can also call it as a satellite DNA. Why it is called satellite DNA? Because it is forming a small peak during density gradient centrifugation. Now what did we discuss? Once again, let us see. We are telling, like, if you want to do the DNA sequencing of two different people to find out who did the crime and all, it's a very expensive task. It's a daunting task. It's a very slow process and it's expensive. Then instead of doing the complete DNA sequencing, if you go with another approach, in other approach, we are just trying to compare the two sets of genes. Instead of complete sequencing, we are trying to compare the differences between the two sets of genes. It's quicker, it's easy. What is that technique called? That's called DNA fingerprinting. So in DNA fingerprinting technique, it involves identifying the differences, not the sequences. Identifying the differences in some special regions. What are those special regions called? What are those special regions of DNA sequences called? They are called repetitive DNA sequences. What do you mean by repetitive DNA sequences? Since they are repeated many times, we call them as repetitive DNA sequences. Now, how can you separate this repetitive DNA? Go with density gradient centrifugation. The genomic DNA forms a big peak. The repetitive DNA forms a small peak. So that is why it's called satellite DNA. Okay, take a screenshot. Right. Now, this satellite DNA, depending upon its base sequence, whether it is AT rich or GC rich, depending upon the length of the nucleotides and depending upon the number of repeats, the satellite DNA is divided into, or we can call them as microsatellites and mini satellites. Let us see what are microsatellites and what are mini satellites. So this, the satellite DNA, depending on AT rich or GC rich sequences, whether it is AT rich sequence or GC rich sequence, or depending on number of nucleotides and depending on number of repeats also, depending on number of repeats also, right? So we can divide the DNA into microsatellites and mini satellites. We can divide it into mini we can divide the DNA into, what did I say? Two types. What are the two types? One is mini satellite DNA. The other one is called micro satellite DNA. Now let us see what are microsatellite DNAs and what are mini satellite DNAs. We told, so one is AT rich, other one is GC rich. Now mini satellites are GC rich sequences. They are GC rich sequences. Then you can tell microsatellites will be AT rich sequences. These are AT rich sequences. First point covered. Mini satellites are GC rich sequences, microsatellites are AT rich sequences. Number of nucleotides. So mini satellites, the length of the mini satellite is 10 to 60 base pairs, whereas the length of the microsatellite is 5 to 10 base pairs only. So the length of this one is 10 to 60 base pairs in length, whereas the length of the microsatellite is 5 to 10 base pairs in length. Understood? The second point also. Now the third point is depending upon the number of repeats. Mini satellites repeat 1000 times, whereas microsatellites will repeat only 5 to 50 times. So mini satellites, they repeat 1000 times, right? Whereas microsatellites, they repeat only 5 to 50 times. So children, I explained you all the 3 points. We are telling that the satellite DNA is of two types, mini satellite, microsatellite. On what basis we have divided them into mini satellite and microsatellite? Depending upon whether it's AT rich or GC rich, depending upon the length, means the number of nucleotides, depending upon the number of repeats in the DNA, how many number of repeats it is. So basing on these 3 points, we have divided into mini satellite and microsatellite. I told mini satellite is GC rich, that means microsatellite will be AT rich. Mini satellite 1000 base pairs repeats. This one is 5 to 10 base pairs in length. This is 10 to 60 base pairs in length. This is 5 to 10 base pairs in length. Number of repeats, if you have to talk, 1000 times it repeats, whereas 5 to 50 times only it repeats. And if you remember the DNA fingerprinting, DNA human genome project, in human genome project, we told that microsatellites are used for genomic mapping. So where do we use microsatellites? Microsatellites are used for genetic mapping. They are used for genetic mapping and they are used for human genome sequencing. In human genome sequencing also we can use the microsatellites, whereas to locate the genetic map also we use microsatellites. And then where do we use mini satellites then? Mini satellites are widely used in DNA fingerprinting technology. So mini satellites are widely used where? In DNA fingerprinting technology. So in the previous board we have seen like who developed the technique, and then what sequences are we trying to look at? Repetitive DNA sequences. How to separate it? How many types of that we have seen, right? It is used in DNA fingerprinting technology. It is used for genetic mapping and human genome sequencing. This is also used for regulation of gene expression also. The second application of mini satellites is it is also used in regulation of gene expression also, children. It is also used for regulation of gene expression. Two important points I told where do we use, and here also I have given 2 important points, and those 3 points also we covered. Quickly take a screenshot of the types of satellite DNA. Yes. Now when we have to talk about, now this whole topic of DNA fingerprinting is about satellite DNA only. Now these satellite DNAs are present in genomic DNA now, they are present in the non-genomic DNA now. They are present in the non-genomic DNA. Means are they coding? They are non-coding. They are non-coding. Means satellite DNAs do not code for any proteins. Means they are non-cistronic and they are repeated large number of times, right? So let us write down satellite DNAs. Do they code for proteins? They don't code. They do not code for any proteins. They do not code for any proteins. Now, if they are not coding for any proteins, that means they are cistronic, non-cistronic, non-cistronic. They are non-cistronic. Now, they are non-cistronic, but they are repeated. But they are repeated large number of times and they make large proportion of the genome. So, they are repeated large number of times. Yeah, we have seen 1000 times it gets repeated. They are repeated large number of times. Since they are repeated large number of times, they make larger proportion of the genome and make larger proportion of the genome. They form the larger proportion of the genome. The satellite DNAs will form the larger proportion of the genome. Now these satellite DNAs, they show high degree of polymorphism. The satellite DNAs even show, what do they show? They show high degree of polymorphism. Now, in simple terms, I'll tell you what is polymorphism. I'm writing my entire lectures with the black only. Suppose one line I wrote with black, another line if I write with red, another line if I write with green, another line if I write with blue. So means I'm changing, right? I'm changing colors, means morphology got changed, appearance got changed. That is morphism. I used 4 colors instead of 1 color. I used 4 colors. You can use different colors also. Poly means many, right? Many, you can see it. That is called DNA polymorphism. Understood? So the DNA is changing in the coding part. DNA is changing in the non-coding part. First itself we told the satellite DNAs are non-cistronic. Non-cistronic means they are non-coding, right? They don't code for, we wrote, they don't code for any protein. Make it clear. If they don't code for any protein, they are not part of cistron then. That means they are non-cistronic. And then we are telling they will just repeat 2, 3, 4 times or they'll repeat 5 to 50 times sometimes 1000 times also they'll repeat. We told they'll repeat 1000 times also. Then that means they are repeating large number of times. If they are repeating large number of times, then they'll make large proportion of the genome. And these satellite DNAs, apart from this, they show high degree of polymorphism also. What is polymorphism? Polymorphism, I mean, I told using different colors is polymorphism. On my lectures, polymorphism of my writing. So in DNA sequences also, if there are changes, then we call it as polymorphism, right? Then what means polymorphism, we tell, is a principle behind DNA fingerprinting. Understood, Amma? Polymorphism, you know, is the principle. It is the principle behind this DNA fingerprinting technique. Is the principle behind DNA fingerprinting technique, and it is also the principle behind genetic mapping also. Do you remember? So polymorphism is also the principle behind. It is also the principle behind, what is that? Genetic mapping. So if you understand what is polymorphism, it will help us to do DNA fingerprinting also, and it will help us to do identify the genetic mapping also. And again, this polymorphic DNA is heritable also. Polymorphic DNA, you know, is heritable. It is heritable, means it can pass on from the parents to the generations, means it can pass on to the offsprings. It's passing on to the next generation. It's passing on to the offsprings. That means 50% of the polymorphic DNA the child will get from the mother. 50% of the polymorphic DNA the child will get from the father, right? Since the polymorphic DNA is heritable and polymorphic DNA is helping us to do DNA fingerprinting, so we can use DNA fingerprinting in paternity testing also. Since polymorphic DNA is heritable, so we can tell DNA fingerprinting. DNA fingerprinting is used. Where is it used? I am telling DNA fingerprinting is used in paternity testing. It is used in paternity testing to find out the genetic father. To find out the genetic father, we can use polymorphism. We can use DNA fingerprinting technology. Understood? So what did we discuss so far? We are just talking about satellite DNAs. Satellite DNAs, they are coding for proteins or they don't code for proteins. They don't code for proteins. That means they're part of cistron or non-cistonic. They are non-cistonic. They are only less number or they are large number. So they will repeat large number of times and they'll make large proportion of the DNA. And this satellite DNA will show variations, polymorphisms. And polymorphism is a principle behind DNA fingerprinting. It is also the principle behind genetic mapping also. Polymorphic DNAs, again, they are heritable. That means they are passed on to the generations. Since polymorphisms are heritable, you can use this DNA fingerprinting technology for finding out the father. Paternity testing, we can use it. Okay. Now, polymorphism. So, what is the reason behind DNA polymorphism? Means mutations, right? So, polymorphic DNA occurs due to, how does it occur? I am telling it occurs due to mutations. Polymorphic DNA occurs due to mutations. We know in mutations, because of physical mutagens or chemical mutagens, there will be changes in the base sequences. That's called mutation. So, mutations will lead to polymorphisms. And mutations can happen in body cells also. Mutations can happen in germ cells also. So, polymorphic DNA due to mutations can be seen in somatic cells also. They can be seen in the reproductive cells, which are called as germ cells also. But the polymorphic DNAs which are formed in somatic cells, you know, somatic cells will not participate in reproduction. So, polymorphic DNAs which are formed in somatic cells are not heritable. So, polymorphic DNAs formed in somatic cells, I am just writing, they are not heritable. Whereas, polymorphic DNA which is present in the germ cells and it is not causing huge variation, it is not impairing, if it is not impairing the reproductive ability. So, polymorphic DNA, the polymorphic DNA occurred due to mutation. It occurred due to mutation where? In the germ cells. In the germ cells, if it is not affecting, the reproductive ability, if it is not affecting the reproducing ability, children, if it is not affecting the means it is not that serious, then it will be passed on to the generations. Then it will be passed on. So, are you understanding? Step by step we are talking about polymorphic DNA because it is a principle behind DNA fingerprinting. We are telling polymorphic, the reason for polymorphism, we told it is mutation. Mutation can happen in body cells also, it can happen in reproductive cells also. Mutation, if it happens in the body cells, it cannot be passed on to the next generations. Mutations which are happening in the germ cells, and if these germ cell mutations are not dangerous, if they are not impacting the reproductive ability, then they are passed on to the next generations. Then you can see them in the population. Understood? Take the screenshot fast. Right? Now, DNA, we are telling. DNA means it is gene. Now, gene, the alternate forms of genes are called as alleles. DNA is changing because of mutation. So, in terms of DNA, can I tell alleles? Can I tell DNA variations means allelic variations can I use? That's what in the NCERT they made a statement with alleles also. They are telling that allelic sequence variations, what are they using? Allelic sequence variations are traditionally called as allelic sequence variations are traditionally, they are traditionally called as DNA polymorphisms. So, we told variations in gene is also polymorphism. So, gene, alternate form of gene is allele. So, then allelic sequence variations is also polymorphism. That's it. Allelic sequence variations are traditionally called as, what are they traditionally called? DNA polymorphisms. It is also called DNA polymorphism. Now, if more than one allelic variation, if more than, what I am writing, one allelic variation. So, this is the chromosome, a pair of homologous chromosomes. Now, on this pair of homologous chromosomes, this is D, a gene, and another D. Capital D I wrote, small d I wrote. So, these capital form of an allele, small form of an allele. So, they are present at the same locus I am showing, right? So, if more than one allelic variation at a locus, I showed the locus. If more than one allelic variation at a locus occurs in a population, where is this present? Occurs in a population. In a population means we have to tell frequency, how much percent of frequency. What are we telling? Allelic, what is allele? This is the allele, and it is showing variation. Where it is showing variation? On the DNA at a location in a population. If showing a variation in a population of a frequency greater than 0.01%, then it is also called polymorphism. Then it is also called DNA polymorphism. Children, be very specific. In NCERT or in the NEET paper, we have statement based questions. We have assertion reason based questions. So, they will give you options and they will ask you identify the correct statement, identify the incorrect statement. So, whenever there are long and lengthy sentences, instead of making your own sentences using your own grammar, try to take the sentence as it is, break the sentence and understand it will be as simple as possible. So, let us see. So, what I am telling, if more than one allelic variations, I showed you an allele. This variations. So, in one population, it is like this. In one person. In another person, it is like this. And in another person, it is like this. On the same chromosome, at the same loci, I showed three combinations, right? Capital D one combination, small d one combination, capital D small d another combination. So, if it occurs in a population at how much percentage? At a frequency of 0.01 percentage, then that is also called DNA polymorphism. Understood? And we are telling if in a heritable, if an inheritable mutation, if an inheritable mutation means the mutation is transmitting, is observed in a population, these mutations, if they are observed in a population, how are they spreading in the population? By reproduction only they are spreading in their population. Now, if they are observed in a population at a very high rate, if they are observed in a population at a very high rate, then it is called as, what is it called? It is called repetitive DNA. So, you are observing that this 0.1% of DNA is appearing in the population at a very high rate, then it is called repetitive. Now don't think that 0.1% only it is repeating. 0.1% only is repeating. In 100, 0.1% means in 100, 0.1% might make a less value. But you know, what is the size of our genome? What is the size of our genome? The size of our genome is 3 into 10 power 9 base pairs, right? The size of our genome is 3 into 10 power 9 base pairs. So if you have to calculate and if you have to keep it, it is 3 billion base pairs. It is 3 billion base pairs. In the starting of the topic, I told 99.9% we are similar. Only 0.1% we are dissimilar, I told. So in NCERT, they asked us, so 0.1% dissimilarity means in terms of bases, tell me how much we are different. In terms of percentage, it is sounding very less. Don't underestimate this 0.1%. They are asking us to calculate and tell us this 0.1% means how much it is. If you just calculate 0.1%, you know it will come as 3 million bases. In 3 billion, 0.1% stands for 3 million bases. That means I am different from the other person with 3 million bases. See how huge I am different. So that is why I am unique. That's why I am unique. Understood? So what did we cover till here? We are just talking about polymorphic DNAs and satellite DNAs. We are telling that polymorphic DNAs, we are just seeing the other ways of explaining the polymorphic DNAs. We are telling if it is an allelic sequence variation, that is also polymorphism. If the allelic variation on a locus in a population, if it occurs at a frequency of 0.01%, that is polymorphism. Now, these are running in the populations because they are heritable and they are repeated. So, these repetitions will help us to identify this polymorphic DNA. This polymorphic DNA vary from one base to many bases. The polymorphic DNAs from one person to other person, they vary from single nucleotide polymorphisms. One base means they are called as single nucleotide polymorphisms. And many, if they are varying by many bases, then that is called multiple nucleotide polymorphisms. Then that is called multiple nucleotide polymorphisms. So what did we discuss in this session is, we discussed everything about what is polymorphism, what is DNA fingerprinting, we discussed. Now in the next session, we will talk about the steps of DNA fingerprinting technology and then the applications, the advantages of DNA fingerprinting technology. If you like the lectures, like, share and subscribe to my channel. Thank you.